wish I could practice at 12 to 12:50, like literally every Monday and Wednesday. I don't know why I did that.
Yeah,
I'm gonna talk to him today, but I do have to leave early today. But
that's okay. I actually just was in here early because I want to figure out how to record the lectures. So for people that are playing sports for practice or for tournaments, if you have to go. So if you need to leave early, that's okay, and I'll have them recorded. So
this is very much like, oh, well, you have to find time to practice.
Yeah, I'm definitely trying to work with all the students because I know there's a lot of student athletes here, and so I'm hoping today it's kind of my trial. We'll see how the recording goes, and then hopefully.
I just didn't want to be like, oh, I'm doing class earlier.
Oh, you're okay. You're okay. Thanks for letting me know.
Yeah. With everything that you have to do, it's gonna be, like, my day is gonna be like 9 hours long. It's terrible. Are you meeting at 7:30? Yeah. No, I don't have a minute. Right. Well, they made it specifically so, well, me and Morales made it specifically so I don't have to, you know. That makes me blissful. So it's like. No, you won't, because there's a lot of snow behind it anyway. No, literally, I'm putting my chair higher.
Wait, can you see? Should I move?
No, it's fine.
Or do you have a, no, do you have a big box? Like, for like,
Or you
charging
drive
it?
USB to USB-C.
My issues and charging.
Oh,
I got the version of it, my phone.
online. That's the one.
I downloaded it.
make sure I'm doing correctly with the science. Are two questions that I cannot determine, like there are two structures.
I started the reading. So I just out of the way. No, we're going.
Oh, and she like files, but there was a way, I know it's like book, like bookshelf or whatever that is.
Also, have you finished, the
I still have like 4 pages to read, like front back.
I read it
She
through.
asked me like an hour ago, she was like, how'd you do it? I was like, I was so she goes, yeah, it's a hard one. I was like, yeah.
I'm not sure it's a challenging one because
Very hard. Like, how
the
many
more I
pages?
read, the more I feel like illiterate.
No, I don't know.
Can I get a full page of notes? I don't know what to discuss. It's not interesting. I don't care
Well,
what.
to an extent, it's interesting, like how this has like two sections. One is like following the paradigm and like producing that experiment out of it, and at the same time, there's some paradigm that is totally changing science or at all. But like that's all I got from the book. Like, how am I supposed to like talk about it? Like,
Right.
even I remember half of the things they were saying.
I don't know who wrote it.
Kuhn.
He wrote it, yeah.
Yes.
I don't like the way he like words.
She likes to use though a lot and like I
Yeah,
feel in a weird
however.
place. However, though,
Yeah.
No.
A lot of that, and that makes it.
She put it in the dryer and I was like, oh,
just give us a PowerPoint of it.
Please. I think he was trying to hit a word down.
Yeah, eventually we'll get there.
Yeah.
she took a bit of it as well.
Or it's always nice to have something challenging.
Yeah.
Not the case. Probably do more.
It was like I tried to get over it. You didn't hear me like I was really dry through, so it was just like. I know, no, just in the middle of the night, you just. Yeah.
I did 10 pages yesterday, and I'll do 10 pages.
I don't know. Well, that's like deeper or anything. Sometimes it sounds like it's kind of.
You can't just.
Oh my gosh, we complimented with possible.
Yeah, that's
I take mine.
even in third year, how to figure out what's best.
And your professor's like letting it, so that's just like.
Easily like disappear and stuff and like reach like 2,000 slides per exam. At the same time, but it's not
We're like,
overwhelming.
I'm on slide 100, that's what I'm like.
Yeah, 1,000.
No, actually. I hope you didn't press all of those.
At some point. I think like that. If I remember everything on the slide, like just the whole stretches. Yeah. That's how I made it. Like survived it. Because
Yeah.
if you don't remember it, just like a stretch. If I remember it, just stretch it.
How dare you?
Alrighty, good morning, everybody. How's everybody doing today? Good. Alright, so I'm gonna start off by just taking attendance. That way I'll be able to learn names a little bit better. And so One thing I want to let you guys know is I am trying to stream this so that it goes onto YouTube at, like, the link will be via YouTube. It'll be on Blackboard for you guys. That way you guys can have like a recording of the lecture. I think it's working at the moment. If I'm unable to get it up there, I'll have like the next lecture up there for sure recorded, but I'm in the process of figuring that out. So it worked a little bit earlier. Right now I'm having a little bit of trouble, but I'm hoping that it will still be streaming and I can just get the URL code for you guys afterwards. So that will make it easier for some of you guys that will have to leave early for practice, or for those that aren't going to be here for one reason or another. So, all right, let me take attendance. Tegan. Lucia.
Here.
Nicolo. Autumn. Ryan. Quinn.
Here.
Celeste.
Here.
Zoe.
Here.
Annika. Jesse. Sarah. Kata.
Here.
Clayton. Paige. Erica. Victoria. Brooke.
Here.
Abby. Hannah. Gabby. Juliana.
Here.
Nicholas. Emily's excused, and then Tyla. All right, awesome. Any questions, comments before we get started with today's lecture? All right, cool. So let's get right into it. So today we're going to be talking about microscopy and how we observe microbial cells. They are microorganisms, so they obviously can't be detected with the human eye, so we need microscopes to help magnify them. And so the suggested readings are these sections in chapter 2 for you guys to complement the lecture material. So, as I've mentioned before, microscopy is essential for understanding microbes and being able to actually get an idea of not only how big they are, how small they are, but looking inwards and seeing the cell structure of certain microbes. And so today we're going to start by understanding a little bit about the physics of light. And so in order to understand how microscopy works, we have to know a little bit about how the physics of light works and the optics of light work, but I'm not going to go into too much detail because I really want to keep this on the microbiology side of things, but it's important to know some of these things as we move forward. And so first we'll start by talking about a simple technique, which is bright field microscopy. It's one of the ones we're going to be using in the lab tomorrow. So it's a nice way to image bacteria. But we're also going to be talking about staining bacterial cells, and we're going to be doing this in lab as well tomorrow. So when you're imaging a bacteria, if you think about it, it's like getting a clear balloon and then filling that balloon up with water and then putting the balloon filled with water into water and then trying to image it. So you have a clear thing inside of a clear thing, and it's like really hard to be able to see, you know, the actual structure of the cell. And so that's why we use different techniques, and we stain cells so that we can be able to have some sort of contrast and see the cell versus its background. So when you have like a colorless bacterial cell in a colorless liquid, you essentially get little to no contrast and you can't see anything. And so that'll kind of, that's one of the analogies I like to think about when I'm thinking of cells. And so sometimes we have fluorescent staining. So they're dyes that can be attached to the cell or certain structures of the cell that help us to peer into the bacterial cell and look at structures within the cell. There are also tricks that can improve the contrast of bright field microscopy, and so some of these are dark field and phase contrast microscopy, and we'll talk a little bit about those. And then if we have structures that are really, really small and we aren't able to see them using some of these methods, then there's some other types that help us to resolve, like super resolution microscopy helps us to resolve structures that are smaller than 200 nanometers, so I'll explain where that number comes from and what that means. It's just an overview. So if we don't limit ourselves to just humans, you'll find microbes everywhere, and so microscopy has revealed that we are just as microbial as we are human, and we live in a very microbial world, and bacteria can be beautiful. So our eyes need help to see microbes. So the tools that we have in order to visualize things are our eyes. And this is just a really simple diagram that's trying to drive home the point that the size at which objects can become visible and be able to distinguish two different objects depends on the resolution of our eyes. So what limits our ability to see microbes is it really comes down to the fact of how the photoreceptors are packed within our retina. So in our eyes, we have our retina and these photoreceptors, and so the distance between the receptors in our eyes is what limits our ability to resolve images and see them. And so our photoreceptors in our eyes are separated by roughly about 0.1 millimeters. And so that's the resolution of our eyes. So if something is 0.1 millimeters in diameter, or how big it is, we're able to see it. But if it's smaller than that, then we need help. So just to drive home this point, resolution is the smallest distance by which two objects can be separated and still be distinguished as two different objects. So to give a little bit of context to how our eyes work, eagles fly high in the sky and they have to see the little things in the land when they're flying above, to get their food and be able to have really good eyes to see those things. So eagles have photoreceptors that are 10 times more packed than ours. And so because they're so much more closely packed, the resolution for their eyes is 0.01 millimeters, so they can see things that are even smaller. And so that's an evolutionary mechanism that's allowed eagles to have better vision than us when it comes to looking at really, really small things from far away. So resolution is just the ability to be able to distinguish two different cells in this case. And so you can see here, you see these two different things. Now, I just want to make sure that we aren't confusing resolution and detection. So detection is just the ability to determine the presence or absence of an object. So it's different from resolution. Here we have a plate and we have some bacterial colonies growing. And so each one of these colonies takes about thousands, maybe millions of cells to make this colony. So you're not seeing the individual cell, you're seeing a colony of cells. So here we have bacteria, I think these are cyanobacteria that are just, spread out, and so you're able to detect them with your eye. You can see the presence of the bacterial colonies or the mold, but you can't resolve the distinctive cells within them. So that's where that comes from. So to resolve most microbial cells, our eyes need assistance, and that's where microscopy comes in. So magnification is being able to increase the object apparent size or the dimensions that helps us to resolve smaller separation between objects. So our eyes need help, we can detect things, but in order to resolve them, we need to magnify them. And that's why we need microscopes. Any questions at this point? All right, awesome. So microbes, they do range in size by orders of magnitude. So this is kind of just to show you the idea of the type of scale, the size scale that we are working with when it comes to microbes. So here we have a Prochlorococcus, and so this is a cyanobacteria, so it's just really, really small, and so you can see the size of it is about 0.4 micrometers. And so here we have Staph aureus. This is of bacteria that exists on our skin, can be a cause of infection, so it's slightly bigger. These are both bacteria, so there are some that are really small, some a little bit bigger. This is E. coli. E. coli is even a little bit more bigger than that at 2.5 micrometers. And then there's Bacillus megaterium. So we're actually going to be working with Staph aureus, E. coli, and Bacillus megaterium in the lab soon. And so just to foreshadow a little bit, Bacillus megaterium, the name mega in here comes from the fact that it's mega, it's big. So that's why it is generally larger. And so when you are looking under a microscope and you're trying to distinguish, for instance, if I give you a tube with both E. coli and Bacillus megaterium, and you're trying to distinguish which is which, well, one, the staining we're going to do is going to stain E. coli pink and this one purple, and so that's one way to differentiate, but also the size is one way to differentiate. Bacillus megaterium will be larger in theory. And so just to put a little bit more of these into perspective, a human red blood cell is about 8 micrometers, so it's a little bit bigger. And then here we have a paramecium. It's just a type of mold, and so this may be just visible to the unaided human eye. So the paramecium is 150 micrometers. And so what I had told you previously is that the distance at which our retinas are separated, or the photoreceptors in our retinas are separated, is 0.1 millimeters. And so 0.1 millimeters would be 100 micrometers. So that's just a little bit of math to remember. I will write this on the board just to keep, so I'm going to be going between millimeters and micrometers back and forth just while I'm explaining it. And I was getting confused when I was going through this, so it's just easier to have it written. So 0.1 millimeters is the distance at which we can resolve images with our eyes. And so this would be 100 micrometers. So you multiply by 1,000 essentially. That's the conversion there. So things that are 100 micrometers or greater can be visible to the unaided human eye. So I'm going to show you just a little video. There's a virus. Viruses are really, really small. We have another virus, influenza virus here. Viruses are typically smaller than bacteria. Here we have a bacteriophage, which is a virus that kills bacteria. And so here we're seeing Staphylococcus, some other bacteria, Lactobacillus, E. coli has its flagella, and then we have a red blood cell, some yeast, a skin cell is 30 micrometers, human sperm cell is 60, pollen is 90, just so very close to what we can see with our human eye. Neurons are just visible to the human eye at about 100 micrometers. And then here are some other things, diatoms and some other larger organisms, amoebas, frog egg. So just to give you guys a little idea of the size scale that we are working with. So, microbial shape. So, like I said, prokaryotic cells are generally simpler than eukaryotic cells. This is there's always exceptions to the rule in science, and so prokaryotes are generally smaller, but there are exceptions, so just important to know that at the back of your mind. So there are different shapes of bacteria that are common to many groups, and sometimes these names will be embedded in the name of the bacteria. Like when I said Enterococcus, the reason it says it's coccus is because it's cocci shaped. So cocci are spheres, and so here's a diagram showing here in the orange. You have these spherical cells, so that's what coccus means. Bacilli are rods. So we have the rods here in purple, and so that's what they look like. Some rods. Spirali are just spirals, so we have them right here. So you have spirali and spirochetes are pretty similar, where they're both spiral, but spirochetes are more flexibly spiral. I think just knowing the spiro or spiri is enough for this class to just know that they are going to be spiral. and some are appendaged, which just means that they have some protrusions. So you can see over here, that's what some of those would look like. But for the class, we're going to be mostly talking about cocci, bacilli, and spirali, or spirochetes. So just to take you back here, to this, generally prokaryotes are the bacteria and the archaea, and they are generally between 0.4 to 10 micrometers. And eukaryotes, the protozoa, algae, and fungi, those range between 10 to 100 micrometers, and 100 micrometers is what's visible to our human eyes. And so some eukaryotes you can see, some of the others you can't. And we even can't see them with the human eye. You would need microscopes and other techniques to be able to look within them and see the actual structures. So it's still not enough. We still need help. So eukaryotes are generally larger than bacteria, but there are exceptions to every rule. So like I said, we are going to be working with some types of, some different shapes of bacteria. You'll also hear me saying a lot about morphology. Morphology just means shape, and so here we are looking at some rods or bacilli. So here we're looking at lactobacillus, two different lactobacillus species, and so you see here they look like rods. This is just a different type of microscopy that was done to show the 3D structure, and it's just really cool to see. Spirochetes, they are spirals, so you can see them like this, and then the bacilli the cocci are spheres right here, so you can see that they're more spherical. So different microscopes are required to resolve different sized scales. So there are a variety of microscopy techniques that allow us to probe the microbial world at different spatial scales. And so here we are going from something that is visible to the human eye, and everything going from left to right is only getting smaller and smaller, and so we'll need different types of microscopy to be able to view things that are smaller than being visible to the human eye. So like I said, the human eye, things that are visible are about 0.1 millimeter, and so that's about 100 micrometers, so that's just about right here. And so things that are things that are smaller than this 0.1 millimeter, we need a light microscope to be able to resolve those images. And so light microscopy goes all the way down to 200 nanometers. That's something to remember, that when you're thinking about what you can see with the human eye, it's important to remember those numbers. And then light microscopy goes down all the way to 200 nanometers. So I will write a little bit of that down. So this is the limit, the human eye resolution limit. And then if we are doing light microscopy, so we're using a microscope that utilizes light to magnify the image, which is what we're using in our labs. So that can go up to about 200 nanometers. So those are the limits of detection. And so, okay. So if we want to see anything in greater detail that's smaller than this 200 nanometers, there's certain properties of light that restrict light microscopy from being able to see things that are smaller, and we'll talk a little bit more about that. But in order to resolve structures that are smaller than 200 nanometers, you do need to use techniques that use electrons instead of light. And so here we have two different types. There's scanning electron microscopy and transmission electron microscopy that fall into that category. We will briefly go through those. We aren't going to be using those in the lab, but it's important to know why they're important. And so basically, the way that these work is we basically coat the sample in heavy metals and then bombard the sample with electrons. And so the way in which the electrons will scatter will give us a topological representation of the surface of those bacterial cells. And so I'll show you a little bit more of what these look like. Anything that is even smaller than that, there are additional techniques, that are used to see things that are even smaller than that. They're really powerful because they don't use electrons or light for detection, like atomic force microscopy, where basically you have this like wickedly small needle that will point and tap the surface of the bacterial cell or the population that it's surfing, and it'll allow it to get resolution all the way down to the pores. And so we're not going to go through all of those in detail, but just to give you an idea, X-ray crystallography, you're really getting a crystallized structure of the DNA or the protein, so that would be really getting down to the molecular level in imaging. So I have a little concept check for you guys. We can walk and talk through this just to gauge, understanding and make sure that you guys really hone down some of these topics. So which of the following statements about the size of microbes is false? A, eukaryotic microbes tend to be larger than 10 millimeters. Prokaryotic microbes are typically smaller than 10 millimeters. This should say micrometers. I thought I changed that this morning, but I don't think it saved properly, so that should say micrometers. Some bacteria are large enough to be seen by the unaided eye, and viruses are always smaller than cells. So yeah, that should say micro for both of those. I apologize. Any ideas as to which of these is false? I'm hearing C. So the correct answer is D. So when you see a statement that says always in biology, that's always a red flag. So like I said, there's always exceptions to every rule. So some viruses are smaller, so that was kind of a trick question there. It was a little bit more difficult, but just to give you an idea of some of the types of questions that could come from exams. So we did say that bacteria are really small and you can't see them by the unaided eye, but when you see things like some, that's kind of one of those things that you would think like, oh, it is possible. There's exceptions to every rule. So this was a more difficult question, but I just wanted to like throw it at you guys and give you an idea. I will try to be a little bit more straightforward when it comes to exam questions, but these are more concepts, and so it's better to get these wrong during class so that you remember those little things, like there are exceptions to rules. All right, any questions? So we know that eukaryotic microbes tend to be larger than 10 micrometers. They range from 10 to 100. And prokaryotic microbes are typically smaller than 10 micrometers. So we have to talk a little bit about the physics of light in order to understand how we're utilizing light to image our microbes. And light microscopy is one of those most powerful tools to study cells. So we're essentially using light microscopes to extend the lens system of our own eyes. So for the light microscopy, we will be using visible light on the electromagnetic spectrum. So the wavelength of visible light is 400 to 750 nanometers. So from this purple violet to this red on the scale. And so for visible light on the spectrum, to be able to observe an object, we have to meet certain conditions. So first, the contrast has to be between the object and the medium has to be at a perfect spot for you to be able to see the bacteria without staining. So remember the balloon analogy that I've said. Basically, if you're trying to image a colorless cell inside of a colorless medium, you're going to get very little to no contrast. So we need to make sure that the contrast is resolved in that case. The wavelength of the light has to be smaller than the wavelength of the object. So the way to think about this is if you put a toothpick, say, in front of a wave of water that's coming to the shoreline, the toothpick hitting the wave of water isn't going to do anything to that wave, right? So it's not going to change the wave or its direction. So if a wave crashes against something large, it's going to change the wave nature of that wave. And so it's similar with waves of light. So you need to have an object that is larger than the wavelength of the light in order for it to change its direction and be able to resolve it. So the wavelength of the light has to be smaller than the object. Does that make sense? Okay. And lastly, magnification helps to spread the light out to improve the resolution to be able to see really tiny things. So the magnification will come from the objective lenses on your microscope. So microscopy takes advantage of how light can interact with matter. So light has both particles and wave properties, and essentially that allows it to interact with matter in different ways. In this case, the matter here is the glass slide. So here we have a glass slide, and there are different ways that light can interact with this glass slide. The first one we're looking at is absorption. So essentially, when you have absorption, you have a glass slide here, and then you have the light, and so the light will hit the glass slide, and that light is absorbed, and as a result, it radiates off heat. And so that's why you have this increase in heat, and so this object is blocking that part of the light because it's hitting it and it's getting absorbed. So does absorption make sense? Just how light hits matter, and then heat increases because that light is absorbed. Reflection is, so light can be reflected, and so I'm sure you guys know this already, but essentially when this wave front of light hits the glass slide, it can bounce off the surface of this slide and can then reflect off into an angle. So there's bouncing of the light off the surface of the slide. So the third one is refraction. So refraction involves the bending of light, and that's really important to remember. So basically, during refraction, the light bends because light travels at a certain speed, and it hits the glass slide here, and the glass slide has a different refractive index. And so we talked a little bit about this last time in lab, but I'll go through a little bit more of what this means. But essentially, with refraction, we have different refractive indexes. So the light will hit the slide, but then it's gonna hit a sample with a different refractive index, and so that changes the angle of the light when it's coming out on the other side. So there's this principle of refraction that we're going to talk about, but essentially it means that the light is going to bend when it enters a substance, and then that will change its speed as it enters and then as it comes out. So this bending of light is what allows a lens to change the magnification in a microscope. So this is the cornerstone of microscopy, and we'll talk about why refraction is one of the important ones that we'll probably discuss a little bit further. And the fourth way that light interacts with matter is scattering. So when you have an object that's really, really small or smaller than the wavelength, instead of having these first three happen, there are certain types of microscopy techniques that allow the light to scatter instead. And so we won't talk about this too much, except that you should know that dark field microscopy is what utilizes the scattering of light. And I'll have this up when I talk about dark field microscopy. But that's just another way that light can hit an object and scatter light. And so here the object size is near, almost pretty close to the wavelength of the light. Any questions on these four types? Okay, so we're going to talk a little bit more about refraction. So magnification requires refraction. So refraction is the bending of light rays. So how does microscopy use this principle of refraction to magnify an image? So here we have a wavefront over here of light. So here we have a glass slide, and then we have this wavefront coming in. And so this wavefront is going to come in and it's going to hit the glass slide. But if you notice, this side, the left side of this wavefront, is hitting the glass slide before the right side of the wavefront is going to hit the slide. So essentially, this part, as it hits the slide, it's going to slow down first, but this other side is still traveling in air. And so this one will get bent first, and this one will then get bent later when it hits the glass slide, because the glass has a different refractive index than air. And so as soon as the light then passes through the glass and then it comes back out into air, you get another bend. And so here, this part will bend first and this part will bend second, because it's going to be traveling still while this one is coming out into the air. So this bending of light is due to having differences in refractive indexes in air versus the glass slide. So here, so how does refraction relate to magnification? Basically, how do we work around this when we use microscopes? So we have our lens, right, in the microscope, and so that lens is curved. And so essentially, let's say that we curve this lens, and so here we have the glass slide, and we're going to curve it. And so essentially, what that does is by curving it, you are, instead of having the light hit at different points and bending at different times, you essentially have the light hitting the glass slide all at the same time. And so now this lens can bend the light rays so that they all go through and intersect at a focal point. And this is the focal point is where you're going to put your sample. So after this focal point, the light rays then can continue on to expand out with an expanding or magnifying of the wavefront, so where it's going to hit the lens. So this expansion will then magnify the image of your sample at this focal plane, and this is the principle of this type of microscopy. So refraction and utilizing these different refraction indices and trying to work around them is what allows the lens to magnify the image for microscopy. Does that make sense?
I would just reverse like the second picture. So like if light's coming in and goes through that glass so that you can see it.
Yeah, exactly. So instead of the light bending, we have these, this is just an example of showing you, like, if we had a bent glass, it's essentially gonna, because the glass is bent and it's not straight, the light will hit all at the same time, and then so basically the light will then converge at a focal point. And so once it hits that focal point, this is where your lens is gonna be. You have your magnification, because we're gonna be looking at it at however many magnification, like if it was 4x or 40x or 100x, and so then that will allow it to expand, and that's how we get an image magnified. Does that answer your question? Okay. Any other questions? All right. So kind of really hit this one on the head, but just to make sure you guys got this, which of the following phenomena allows a lens to magnify an image? Refraction. Good. Allows us to bend the light towards the focal point where we put our specimen, and that focal point is where the wavefront can expand and magnify. All right. Let's see what time it is. Okay. So we're going to get into some of these techniques. The first one is bright field microscopy. And this is the most common type of light microscopy because of its ease of use. So essentially, some of the things to note when we are doing bright field microscopy is it's generating a dark image of an object over a light background. So that's where you get this bright field name, because you have this bright field around it that you're trying to utilize to be able to look at your image, and so it changes the contrast of your image. So this is one of those where I talked about the balloon situation, where if you have a clear cell or a clear balloon and you put it in a clear liquid, so if the cells are in water, that's essentially going to make them really hard to see. So you want to start by using bright field microscopy, and in order to make sure you increase the resolution and be able to see the cells, one, you need to make sure that you use a shorter wavelength of light. Two, you want to make sure that you balance the amount of light that you're using with the contrast. So too much light can be too intense, and you'll see on your microscopes that dial where you can increase and decrease the intensity of light. You can play around with that to see what looks best when you are using them. We also use immersion oil in this case, and so the point of this, and we'll talk a little bit more about it next, is it helps to take advantage of differences in refractive indexes. So immersion oil has the same refractive index as glass. So that's why we don't have that bending of the light and refraction. And lastly, in bright field microscopy, if you want to see these little tiny cells clearly, you want to use the widest lens. So we usually are able to visualize them at the 100x lens plus the oil. So, use of immersion oil in bright field microscopy. The refractive index is this ratio of the speed of light in a vacuum to its speed in another medium, so that can be air, water, glass. And so the refractive index of the immersion oil is 1.5, which is the same as glass. And so that's why over here, if you see here, we have our lens, objective lens, and this is our glass slide, and then here's the focal point in the center. And so if we look at this side where we don't have any oil, you get the light coming in. It's gonna go through the lens and wait, where are we looking at? Sorry, no, we're looking at this one. So if we're looking at the red side, we have the light coming in from the red. So we have it coming in from here, and then it's gonna get bent, and you're not gonna get the light going through the lens so that you can see it well. You're gonna lose a lot of that light. When we put immersion oil, if you follow the blue path, the immersion oil has the same refractive index as the glass slide, so we don't lose any light, and you get the light source coming up through the lens, and you get to see your image a lot more clearly. So it minimizes the loss of light by refraction, and it makes 100x magnification possible without distorting the image too much, because we usually use immersion oil only at the 100x lens. Any questions at this point? All right, I'm going to show you something. So we have these glass beakers, and we're putting immersion oil in there. And so you can see that because the refractive index of immersion oil in glass is essentially the same, when you keep pouring the immersion oil, the inner glass disappears and you can't really see it. So that's kind of a cool way to remember why we are able to see images better when we use immersion oil. So in lab, you guys will use a compound microscope. So that's the microscopes that you saw already, and it's a system of multiple lenses designed to correct or compensate for aberration. Fancy way of saying what a compound microscope is. So the objective lens, which are these four lenses, the 4x, the 10x, the 40x, and the 100x that we've talked about before, those are the objective lenses. Those are the first step of magnification. Like I said previously, the ocular lens, which is the eyepiece, that's a second type of magnification. So within the eyepiece, we have 10 times magnification. So the total magnification is magnification of the objective lens multiplied by that of the ocular lens. So if we have an objective lens, at 40x and the ocular lens is at 10x, and we're looking at our image through this 40x lens, what's the total magnification for looking at that specimen? 400, yeah. Cool. So the reason it's called a compound microscope is just because it's compounded by multiple objective lenses. So just to remember that a little bit better. So, preparing your specimen for microscopy. Tomorrow you guys will be doing a wet mount preparation, and it's essentially a simple way to observe microbes by placing them in a drop of water on a slide with a cover slip. So there are advantages, and this is gonna also be a little bit of a repeat of what I've talked about in lab last week, but we are gonna be doing this tomorrow. So some advantages of doing a wet mount preparation is you can observe the cells in a natural state. So they're living, they're moving, they can be fast. Some disadvantages are that there could be little contrast between the cell and the background. So that's that whole balloon analogy again. Some microbes move really fast, and then the sample can dry out quickly because the specimen is in a water drop, and then you put a cover slip on top to sandwich it and make sure that it's, you know, sticks to the glass, but it can also dry out quickly. So when you're going to be finding focus in your microscope to be able to observe some of these samples, bacteria are observed at different focus levels. So here you can see that you have this like really blurry image, and so essentially you're too far beyond the focal point. And this will make more sense when you're actually looking through your microscope and moving the fine knob, the coarse and the fine knobs, to be able to like magnify and demagnify your image. So sometimes if you go like too far and you like twist the fine knob a little too much, you start to see something like this. If you are too close to the lens, you can see something that's a little bit more blurred. And then something that would be in focus, basically you're looking for something that looks a little bit more sharp. And so this is just to show you guys an example of what you're looking for under the microscope. And so here this is a spire sheet, so you would want it to look something like that when you're getting it in focus. Sometimes when we're doing wet mounts, so these specimens, these bacteria are living, they're moving, so they're not always going to be happily laid on their side to where you can see them perfectly like this. Sometimes they are like sitting upwards, or sometimes there's a couple of them on top of each other. So there's going to be layers that you can see the image with. There's no like one perfect standard way. And so when you're looking through your microscope, if you see something like this, then it's probably standing up, it's probably not laying flat, and so that's probably why. And so you can move around your image and find cells that are more easy to detect. All right. So, fixation and staining. Let's see. Yeah, we got time. So, the resolution and the contrast of cells can be enhanced by fixing the cells to the slide and staining. So these are a different type of microscopy that isn't the wet mount preparation, because when you stain and you fix cells, you are killing the cells. So fixation just means that the cells are made to adhere to the slide in a fixed position. And this could be done by methanol or heat flaming. The technique we'll use in lab is we'll heat flame the slide to make sure the cells are fixed to it. And then we can also stain, which gives the cell the distinct color. And so the way that this works is that the stain is a positive charge, and the cells have a cell envelope that's a negative charge. And so the stain is able to bind to the negative charge of the cell envelope of the cell. And so that's how it gets in there. So both methods usually kill the cells. So if you want to see live cells, you won't be doing this. So a simple stain is what you're going to be doing in lab tomorrow. So we're going to do a wet mount, and then we're going to do a simple stain. And what it does is it adds dark color specifically to the cells, but not to the external media or the surrounding tissue. Methylene blue is the commonly used stain, but in the lab tomorrow we're going to be using a crystal violet stain and we're going to be staining E. coli. So the procedure, the way that this works is we'll have tubes of E. coli, and you're going to have a tube or a loop in your drawers, and so you'll grab some E. coli. We'll do this aseptically, so I'll talk about that tomorrow, but we'll essentially place a loop full of the E. coli culture onto a clean slide, and we'll spread it over. Make sure we get a good surface area of the glass slide with the E. coli spread on there. And then we'll allow it to air dry a little bit because we don't want to heat fix it, which is the next step, when it's still wet. So we'll let it air dry, and then we'll fix the cells to the slide. So we're not going to be adding a drop of methanol, we're going to be heat fixing it. So I'll talk about what that means tomorrow, or how to do that tomorrow. But it's essentially just running the slide through the flame very quickly to make sure that the cells stick on there without killing them or completely removing everything that's on there. Then when we heat fix, we'll let the slide air dry, just let the cells cool just a little bit, and then we'll stain. So we'll stain with crystal violet tomorrow, not methylene blue. And then we'll leave the stain on there for about 30 seconds to a minute, make sure it gets into the cell walls. So when we heat fix, we're also allowing the cell to pop open and break open, and we'll have access for the cell wall, which is negatively charged. And so when we put the stain, which is positively charged, it's able to actually get in there. And then we'll wash the stain off with water, and we'll blot off any excess water. We'll have blotting paper, and then it's ready to look at under the microscope. So we're going to be doing this week, tomorrow, and then next week we're going to do a differential stain, which is a little bit more complex, but it incorporates the same principles, where you're going to have, in a simple stain, you have one organism that you're trying to stain. In a differential stain, you have to differentiate between organisms, so you're going to have more than one organism. So we'll have 2 or 3, but we'll start with 2, I think, 2 or 3. But the most famous one, this differential stain, is the Gram stain. So it usually targets the cell wall structure of gram-positive versus gram-negative bacteria. Have you guys done any Gram stain before? No? Okay, cool. So essentially, gram-negative bacteria will be stained pink, and gram-positive bacteria will be stained purple. And I'll explain this. So the Gram stain was devised in the 1800s by the Dutch physician Hans Christian Gram. And so the way that this works is, over here I'm showing you these cells from a patient's tissue, and so over here we have some, ignore these pink ones, these are just some of the tissue that's stained, but the cells here that are causing the infection are these purple cells. So these are gram-positive bacteria because they retain the crystal violet stain because they have a thicker cell wall. And this will make sense in the next slide. So the gram-negative bacteria won't retain the crystal violet, and so that's why the gram-negative turn out pink and the gram-positive cells turn out purple. So we're going to go through the procedure. All right, so over here we're looking at the color of the cells, and over here in B, we're looking at the differences in the cell membrane of gram-positive versus gram-negative cells. So just to keep things straight, I'll have both of these up as I talk about it, and next week I will definitely go into more detail of the differences in the cell walls of gram-positive and gram-negative cells, but for today, I just want to drive home the Gram stain procedure, and then I will just close the door because it's getting a little bit loud out there. Would you mind closing the door on your way out, just because it's getting loud? Thank you. All right. So, in the Gram stain procedure, you have your gram-positive cells and your gram-negative cells, and they're both clear. They both start off as clear. We haven't put anything on them, but we fixed them to the slide. So the gram-positive cells have this thick peptidoglycan layer, so that's just what the cell wall is made up of, and I'll talk about that next week more in detail, but for now you just need to know that the gram-positive cells have this really thick peptidoglycan layer. This is the cell membrane here. And the gram-negative cells have a very thin peptidoglycan layer here shown in blue, but that is also, they have the cell membrane, but then because the peptidoglycan layer is really thin, these gram-negative cells have an outer membrane that coats the peptidoglycan layer just because it's so thin. So we add crystal violet as our first step in the gram stain procedure. So the crystal violet stains the cells reversibly. And so what this means is that it can be washed out. So we add the crystal violet, and that gets into the cell walls. So here we see the crystal violet has positive charge. These cell walls, the peptidoglycan layer has some negative charges. So we see these cells turn purple if we were to look at them after this step. So the next step in the gram stain procedure is to add iodine, which binds the stain to the gram-positive cells. So the iodine essentially creates this complex with the crystal violet. So it makes sure that the stain is no longer reversible in the gram-positive scenario. So iodine and crystal violet just form this complex to really adhere the stain in the cell wall. Make sure it just gets in there and it stays. So at this point, you see that the cells are still purple because the iodine doesn't do anything. We've added the crystal violet. But next, we then wash with a decolorizer. So in this case, we are using ethanol. And so that's going to decolorize the cells. So in the case of gram-positive cells, the iodine and crystal violet complex, because there is this really thick cell wall that helps to capture the crystal violet dye, and it stays in there. But because the gram-negative cells have these thin cell walls, then the stain is removed from the cells. So the iodine complex with the crystal violet doesn't do anything to the gram-negative cells, and the stain can be washed off. So this is where the difference in the cell wall comes in, and this is why it's a differential stain. So the last step is you add safranin counterstain. So safranin is used because it's just a lighter stain than the dark purple crystal violet. It's pink. And so I don't know who came up with using pink and purple for stains because they are very close in color when you're looking under the microscope. But that's just generally what we use in gram staining. And so in this case, when you add the safranin or the counterstain, the gram-negative cells have lost the color. So the pink stain, this counterstain, can get in there. But if you're adding this lighter pink stain on top of these gram-positive cells that are now purple, the pink isn't strong enough to override the purple. So that's why the gram-positive cells remain. They absorb the safranin, but they remain the dark purple. And then the safranin counterstains the gram-negative cells, and so those will look pink when we look at them. So just an FYI, the stain is pink, but for people that have trouble seeing colors, the majority of people, those that are colorblind or just in general have issues, we have a Bismarck brown stain, and so that's the counter stain that we use as an alternative. Some people just use it because it's easier to see than the pink. So it's just something to know that there are other counter stains, but the general one that we use is safranin, and that one is pink. Any questions on this? All right. So other useful stains are, so one of them is a negative stain. So basically what it means is it's just coloring the background instead of the actual cells. So it helps to make the capsules and the cells more visible. And then we have spore stains. So fungi, some fungi can produce spores. And so it's actually pretty cool. We'll probably do the stain in the lab at some point where we have this malachite green stain that stains the spores green, and then the actual cell is stained red, and so you can see the spores within the red cells, and so you can be able to differentiate that and be able to detect fungi that way. So quick concept check. What is the correct order of reagents in the gram stain procedure? So we can use process of elimination here to start. What's the first stain that we use? The crystal violet, so B or C. And then we either then use the iodine or the ethanol. So the iodine is creating that complex that keeps the crystal violet locked in place. And the ethanol is our decolorizer. So it's really important to know, like if I ask you on an exam, what's the decolorizing step? You should know that that's the ethanol. And so B would be the correct answer because we put the crystal violet, we lock it in place, then we decolorize, but the crystal violet is locked in place in the gram-positive cells, but not in the gram-negative cells. And then we use the counter stain, which is safranin at the end. So the answer here is B. Make sense? All right. So for the rest of this time, I'm just going to quickly go through some other types of microscopy, just so you have it, just so you know. But these won't be things that we'll use phase contrast microscopy in the lab, but we won't be using dark field. But just to drive home this point, there are cellular structures that are difficult or even impossible to detect with bright field microscopy. Either they're really small, below the limit of resolution of light, which is what we're able to resolve using light microscopes, or they're too clear and the cytoplasm is transparent. So dark field and phase contrast microscopy take advantage of special properties of the light waves to get around these issues. Now remember the 4 different types of properties of light waves, which were absorption, reflection, refraction, and light scattering. So dark field uses light scattering and phase contrast utilizes refraction. So the thing to remember here is that dark field will utilize scattered light to be able to detect things. So the specimens here are visualized as these really bright halos of light around a field of darkness. So you can detect really narrow cells that are unresolved by bright field microscopy. Here's some more images. So here you can see that these specimens are really like these bright halos. And so in phase contrast, it exploits differences in the refractive index between the cytoplasm and the surrounding medium. So this amplifies this difference in the contrast between the cells and the actual media. So even cells that are very clear will look much darker. And so you don't need to know any optics stuff here, but just knowing some of these key terms, that's where this comes in. So differential interference contrast microscopy, or DIC. This is just one of the last types of light microscopy that I want to talk about quickly. It enhances the contrast falsely. So these are 2 images of the same thing, but essentially here we have this false 3D effect. So it enhances the contrast by superimposing an image of the specimen onto a second beam of light that generates interference fringes. Provides depth by generating a false 3D effect. So basically what it's doing is that it's taking 2 images of the same thing and it's putting them on top of each other because of just having like different light. If there's like a second beam of light that's also targeting that same thing, and so it puts those 2 images on top of each other, slightly staggered, so it's superimposing them. And that will give you this false 3D effect to be able to see. So here, these are the cells, looking at them under just regular phase contrast and then DIC. So here's a quick concept check. And so this is one of those questions that really what I'm doing here is I'm unifying concepts of phase contrast microscopy from 2 different slides. So this is kind of an example of the types of questions I would ask on an exam where I'm asking you something about phase contrast microscopy, but I talked about it in 2 different slides, so you just want to unify those concepts. So Yeah. So which of the following is true about phase contrast microscopy? It exploits differences in refractive indices between cell parts and the surrounding medium. I just told you that, so that one is true. It can be used to view live cells. Is that true? Yeah, I see some nods. Yeah, so we use phase contrast microscopy for the wet mounts. Remember, we're gonna be doing that tomorrow. Phase contrast, wet mount, they're gonna be live, they're gonna be moving. If I were to ask if we're doing bright field microscopy at 100x plus the oil, we're gonna be looking at dead cells. Only used for specific growth phase of the cell. That is not true. I didn't talk about anything regarding growth phase, so the answer is D, both A and B. Any questions at this point? Okay. So the last really quick couple of things are, I just want to quickly talk about fluorescence microscopy. So in fluorescence microscopy, basically the specimen absorbs light of a defined wavelength and then emits the light of a lower energy, so it's a longer wavelength. So I'll talk about what that means. So you can use this to view cell processes in various cell types. But essentially, what fluorescence microscopy does is, and what you guys really need to know, is that we're using a fluorophore or a dye, so that will fluoresce. So this fluorophore or dye will absorb the light at a certain wavelength, and then it's able to emit that light by fluorescing, so the specimen can fluoresce. And so there's a fluorophore that's absorbing the light at a wavelength, and then it's emitting fluorescence, and that emission is energy. So it's emitting fluorescence through energy. So the thing to remember here is that the longer the wavelength, the lower the energy that's being emitted. So energy and wavelength, or fluorescence and wavelength, are inversely proportional in this case. So the longer the wavelength that it's absorbing the light at, it's going to emit it at a lower energy, and then vice versa. So green fluorescent protein, or GFP, is one of those dyes that was one of the first really that was developed that can be used to fluoresce cells, and so it fluoresces as green. And it's interesting because it can be expressed from the DNA of almost any organism. So here we see all of these organisms fluorescing green. And so we have also created subtle mutations in this green fluorescent protein to be able to give us a palette of different colors that we can use in the same experiment to fluoresce different structures and label different structures within the cell. So we are able to label structures within the cell with fluorescent proteins, and that will emit a fluorescent image. So concept check. In fluorescence microscopy, the specimen absorbs light, and then it emits it at a blank energy and thus a blank wavelength. So how are energy and wavelength here related, I guess, is the question I'm asking. How many of you guys think A? B? All right, yeah. The answer is B. So good job, you guys. That's one of those things that's kind of, you just got to remember. So if you have something at a lower energy, you're going to have a longer wavelength. I briefly talked about electron microscopy. I just have this slide and then one other one. So basically, instead of using light, we're using electrons, and these electrons are behaving like light waves. So they're very high frequency, and they allow for really, really superior resolution. And so the sample is coated with a heavy metal, and then the sample has electrons that are shot at it, and so it has to absorb those electrons. So there's this electron beam, and the sample, they're in a vacuum. And then the lenses here act like magnetic fields, so there's no light. And two types that are pretty cool are TEM and SEM, which are transmission electron microscopy and scanning electron microscopy. So you coat the specimen with the heavy metal and then you shoot electrons at it. And so in transmission electron microscopy, here the cells require slicing. So you have to slice the cells first, and so then the electrons can pass through the specimen because you've sliced them, and so you can look at internal structures. And with scanning electron microscopy, the electrons then just scan the surface of the specimen, and it reveals the external features in the body. So when we use transmission electron microscopy, one, we're slicing, which allows the electrons to pass through, and two, it reveals internal structures. With scanning, we're scanning the outside surface, and we're not slicing, and you can see 3D features of the external structure. One thing I'll note is with transmission electron microscopy, the electrons have to pass through the specimen. So if there's like a lot of like here in the dark areas, it's probably like thicker. So the electrons will pass through like more slowly, or they're not able to pass through as much. So when we have like more white inside of the cytoplasm, it's easier for the electrons to pass through. You don't really need to know any other optics of any of the other types of microscopy except for light microscopy, and so these are just some other types like electron microscopy, and I just expect you guys to know these key points. Any questions? Yeah.
For the last concept check, I know you said like the energy and the wavelength were inversely related, and I get how like B is right. Wouldn't C also be right?
So the specimen is in the room, let's say at a. Yeah, but the way, so it's essentially, yeah, in theory, like the inversely proportional concept is right there, but in fluorescence microscopy, we always want the wavelength to be longer of the actual specimen, because remember, if we have, if the specimen is like, if we have like a toothpick and it's hitting a wave, it's not going to do anything, so it has to be longer than the actual wave, the light wave. And so that's why we go with B, because we have a longer wavelength of the actual specimen than the light. And so this, it's going to emit it at a lower energy. So that's kind of one of those principles of just standard like chemistry and physics, where things that will require lower energy are going to be more favorable. And so yeah, that's a good point though. That's a good observation. So does that make sense? Yeah. All right. Any other questions? Okay. So I'm hoping that the streaming and recording worked. I will put those up on Blackboard for you guys. If not, I apologize, and I will definitely have it figured out for next week. We do have lab tomorrow, so I will see you guys tomorrow either at 10 or at 1. And yeah, I will see you guys then.
What's up?